Everyone, I hope you are fine. Today we are talking about the chemical reactions inside the living organisms, and we said or talked about before this type of reactions, and we said every day that we have a metabolism that occurs inside your body, and these two processes of metabolism may be catabolism and anabolism. We said that catabolism is a breaking down of the food from complex form into simplest form, from complex molecules, from macromolecules into simple molecules, and this process here produces energy. And we said before that anabolism is a type of the reactions that living organism cells that done to collect the micromolecules to form macromolecules, and this process is need energy. So first that occur catabolism and then anabolism. So catabolism occurs first to produce energy, and then the anabolism use this energy to form macromolecules. And we said before that the importance of these metabolism reactions, we use it for growth of the body, for repairing the damaged tissue, and these two types of reactions is very important to the body, as anabolism for growth and repairing, and the catabolism for obtaining energy when breaking down the complex food. Here are the enzymes. Of course, enzymes are very important. Chapter 3 here talking about the enzymes and talking about the activation energy. What is the activation energy? It is the minimum energy that needed by the body, minimum energy that needed by the cell to do all the chemical reactions or to do all biochemical reactions. So here the activation energy is what? Minimum energy that needed by the cell to do all chemical reactions. So here the cell needed 38 ATP. That means it's the minimum energy that needs by the cell to do its activity. But here, guys, each cell needs energy. How to decrease this energy? How to make this energy decrease and not to consume all this energy? So we can use enzyme. So the effect of the enzyme on the activation energy is to decrease the activation energy that needed by the cell. So here, what is the difference between two graphs here? Here, the violet one, it's activation energy of uncatalyzed reaction. Yes, here in the violet line, we can't use the enzyme. But in the green line, of course, here we use what? Yeah, of course, we use enzyme. So difference here, there's a violet one with enzyme, but the green one, the violet one without any enzyme, but the green one with enzyme. So the enzyme have a big effect on the activation energy as it decreases the activation energy that needs by the cell. We said that we have activation energy, and this activation energy may be decreased by the enzyme. So the importance of enzyme here, we revise it quickly as we discussed it before in the last video, that it decreases the activation energy and it decreases the consumed energy by the cell. Also, it makes the reaction occurring quickly. The properties, or before the properties structure of the enzyme, we said that the enzyme is made up of amino acids. It's mainly made up of, proteins. So it's made up of proteins or amino acids. Each enzyme have active site. So the enzyme, its origin of the enzyme or the word or enzyme comes from that it make activation or activator of the cells for the chemical reaction. Properties of enzyme. The first property of enzyme is similar to the catalyst, as the enzyme speeds up chemical reaction, and also the more specific as the enzyme more specific for the reactant, for the chemical reactions, and also for it reduces the activation energy that's needed by the cell. Also from the properties of the enzyme that, we discussed before that is important, we're affected by temperature and pH of the reaction. So if the temperature is become not suitable for the enzyme, it will not work. Here the enzyme is more specific. Why? Because it have active site here. Look, the substrates enter to the enzyme as a key and lock, as we said before, as a key and lock, and produce enzyme substrate complex here intermediate. And the enzyme reacts with substrate to produce what, guys? Yeah, product and the enzyme comes out from the reaction as it is without change in structure and properties. Here we are talking about the factors that affect the speed of chemical enzyme reaction. What are the factors? Yeah, temperature. Temperature is the first factor that affects on the enzyme action. Why? Because we have something called optimal temperature. You know that the human body temperature is what? From 37 to 40, yes? Of course, from 37 to 40, and it's a range or thermal range for the enzyme to work inside your body. As we saw, the enzyme, it's inside the living organisms. So the enzyme, the temperature for the human, or the enzyme activity in the human, its thermal range from 37 to 40. And the optimal temperature, optimal what? Temperature. What is the optimal temperature? It is the temperature which is the enzyme is more active. Here the temperature is more effective. Why? Look, if the enzyme here, Ms. Dua, is decreased below the optimal temperature, for example, at 35. Maybe in the winter that your temperature is what? 35 or 36, yes? Yeah. You can't do any activity. You can't move. Why? Because the enzyme not work. As it below the optimal temperature. It below the optimal temperature, yes. So the enzyme it not work well. So it need what? It after time if increase the temperature or warm up, it will restore again its activity or reactivate again its activity. So when the enzyme decrease, then the optimal temperature, what happened? It stopped? No, it decreased its activity until it stopped, and it may restore again or reactivate it again after rising temperature. But if the temperature is more than, yeah, it's above the optimal temperature, what happened, Above 38, above 40. You range from 37 to 40. Above 40, of course. The enzyme is protein in nature, so the protein will damage and the enzyme will not work and it denaturated and not reactivate again because it denaturates die. Okay. Here is the enzyme minimal temperature. It's the temperature which is the enzyme is less active one, which appears in the graph here. It's optimum temperature here, and here is the minimal temperature. Above the optimal temperature here, what happened to the enzyme? It denaturated directly and die, and the enzymes will not work. The temperature, we can use it in detergents. Yes, some detergents like, the Oxy detergents, the Persil. Yes, they are enzymes, they act as enzymes. So if we increase, then the optimal temperature, these enzymes will not work. Okay, and denaturate it. Okay, guys, here we are talking about the enzyme and its activity with temperature. What is the difference here between the two enzymes here? Enzyme A and enzyme B. Here is the thermal range of enzyme A from what to what? Of course, from 15 to 55 for A and B. And the optimal temperature here is the same? Yes, not the same. As here in enzyme A, the optimal temperature is 35, but here in enzyme B, look, maybe 42? Yeah. So they are different here, okay? So here the thermal range from 16 to 55, but the optimal temperature for each one is different. pH. What is the pH? The second factor that affects the enzyme action. It is a concentration of the hydrogen inside the solution here. We have a pH line, and this pH line indicates if the solution here or the material here is acid or base. If the pH less than 7, that means it's acid solution. If the pH more than 7, the concentration of hydrogen ion more than 7, that means it's basic solution. But if equals 7, it's a neutral, not acid, not base. So here, Ms. Dua, the acidic is red and the basic is blue, and the neutral not acid nor base. Okay, so it's according to the pH line, yes. How the related the enzyme activities with pH? Yes, of course, some enzymes in nature, the enzyme is protein, consists of what? Amino group, super, and carboxyl group. Is acid and base? Yeah. So most of enzyme inside your body is neutral, as they are protein. Yes, and they have amino group and acidic group, of course. So they are worked at neutral medium. But some enzymes may be worked in acidic medium, like pepsin. You know the pepsin? What is the pepsin, Ms. Dua? Pepsin is the enzyme that's used, look here, I'm sorry guys. Yeah, here we said that the enzyme activity is affected by the pH value as it contains acetic acid carboxyl group COH and the amino group NH2. And we have optimum pH. Optimum pH is the pH which is the enzyme is more effective, more efficiency, okay? And we have some enzymes that work it at acidic medium like pepsin, and some that like pepsin that work it in the stomach, and some enzyme work it at alkaline medium like trypsin that work in the small intestine. But most of enzyme inside your body is neutral effect. Why is it neutral? Because it contains what? Yeah, acidic group and carboxyl group. Acidic group and carboxyl. So the most of here is what is neutral. Is what you is neutral. Okay. Here we have finished the factors that affect the enzyme. I hope you are fine and studied this part well and discuss it again in the Zoom session. Thank you. Goodbye.